Okay, Aparna single, right?
Yeah, yeah, single.
Okay, Aparna, are you feeling comfortable?
Yeah, yeah, yes, sir.
So ready for your mock interview?
Yes, sir.
Okay, Aparna. So introduce yourself in a brief.
So, my name is Aparna Singhal. I am from Meerut, Uttar Pradesh. I have done my bachelor's in biotechnology from Chaudhry Charan Singh University, Meerut. After that, I appeared for IIT JAM, in which I secured All India rank 16. After which, I got an opportunity to study master's in biotechnology from IIT Bombay. During campus placement, I got recruited by an edtech from Byju's, wherein after MSc, I started to work. My major area of work was content creation in biology and just a few live classes also, as and when required. After the notification of post of examiners of designs and trademark, I resigned my job and started to dedicate my full time for the preparation of this exam.
Okay, so you have done
Like
what in.
past August only. Yeah, past August only I have resigned and I have a 3 years of corporate experience.
So right now you are preparing on government examinations, right?
Yeah, right now I just focused on this exam only because I was like there was a transition of corporate to the that of the studies, no, so it was a bit hard for me to just accommodate and adapt for the study. Now since I'm adapted, so I'll continue to give more and more exams now.
Okay, so you did your MSc from IIT Bombay, MTech or
Yeah,
MSc?
yeah. MSc IIT JAM I appeared, so that's why
Okay,
MSc.
MSc in biotechnology.
Yeah.
Okay, so what is your area of interest, Aparna?
So it's like my area of interest are like 2 to 3 subjects I have prepared: molecular biology, recombinant DNA technology, and immunology. And I have studied a bit of every subject, but not sure how much I'll be able to like
Okay, that
recollect right.
that okay, okay. That is, you are more confident on molecular biology, recombinant technology, and immunology.
Yeah, yeah,
Okay, so can we have some questions of regarding molecular identity and immunology?
yeah, yeah. Sure.
Okay, start from basics. What is the difference between nucleotide and nucleoside?
So nucleotide is composed of nucleosides as well as phosphate groups, whereas nucleoside is just like sugar molecule. Nucleotide is nucleoside plus phosphate group, whereas nucleoside is sugar plus that nitrogenous basis.
What are the different chemical bonds involved in the formation of a nucleotide?
So number first I remember is like the phosphodiester bond that is present between the phosphate groups and the sugar moiety. Another is if I consider the DNA, then of course the hydrogen bonds between the nitrogenous bases. These are the
No,
two
no, I
bonds
am
that I
talking just only about the single nucleotide. Suppose you have ATP molecules. So, what
Mhm.
different kinds of bonds are there in a single nucleotide?
So it is majorly phosphodiester bond, glycosidic bond.
Okay, generally we use 3 prime, 5 prime directions or reading sense in the DNA reference. What is the meaning of these 3 and 5 prime?
So it refers to the free groups. Like when we see there is 5 dash free phosphate group at one end and the three or hydroxyl group at the other end. That's why we read that way. And the reason behind lies in the polymerization with the help of DNA polymers. Basically, DNA polymerase polymerizes in 5 prime to 3 prime. That's
3
why
number or 3 position, third position,
position of carbon.
carbohydrate, that means sugar moieties.
Yeah, sugar moieties. One is for hydroxyl group and another one is for free phosphate group.
Okay,
Yeah.
very good. What is gene expressions, Aparna?
So gene expression is basically whatever information is there in the DNA that needs to be encoded. So gene expression is the process by which the information in a DNA is encoded into a protein. So it can be like RNA, like DNA to RNA to protein. So this is the central dogma that actually describes the gene expression phenomenon.
Okay, means the whole process from the DNA to the protein that will
Yeah,
consider
yeah,
the gene
functional protein.
expressions.
Yeah, yeah, functional protein. Yeah,
Okay, in prokaryotic cells, that means bacteria, there are certain reported operon, right, for the
yeah, yeah.
expressions of a gene, certain genes. What is the meaning of
Mhm.
operon?
Operons are basically a group of clusters of a gene that are like tightly regulated and are transcribed together. For example, there are few components of operon, promoter sequences, regulatory sequences, or the structural genes that are transcribed together to give rise to a certain product, like certain proteins, basically functional proteins. If I were to tell you example, then lac-operon and the tryptophan operon are the two most prominent examples in bacteria, and both have their own characteristics upon which a certain protein is expressed, which gives bacteria ability to survive and grow.
Well done. Aparna, have you heard about the anabolic and catabolic operon?
Yeah. So, as the name suggests, catabolic is degradation of something, and the anabolic is synthesis of something. So, similarly, lac-operon is an example of the catabolic operon because therein beta galactosidase is produced, which has a main role in degradation of lactose to glucose and galactose. Whereas anabolic is when there is a synthesis. So there are five structural genes in the tryptophan operon E, A, B, C, D, E. So they code for an enzyme that together result in synthesis of tryptophan. So tryptophan is an example of anabolic operon, and lac operon is an example of catabolic operon.
Okay, Aparna, suppose if I use the term that is RNA dependent DNA polymerase, what is the mean of that?
So RNA dependent DNA polymerase means a DNA polymerase is basically the enzyme that polymerizes the reaction. But when it uses the template as RNA, then it will be RNA dependent DNA polymerase.
So, what will the function of that polymerase that function as a RNA dependent DNA polymerase?
I think the example of that will be reverse trans. No, that will be using RNA. So can you repeat your question once? I just.
Okay, my statement was that is RNA dependent DNA polymerase. What is the meaning of that?
So I told you just the polymerase enzyme, the DNA polymerase enzyme that will be using RNA as a template.
And what is the meaning of a DNA dependent DNA polymerase?
DNA dependent DNA polymerase is the enzyme that will make use of DNA to polymerize the reaction.
And what about the DNA dependent RNA polymerase?
DNA dependent RNA polymerase is again okay. So I'm just I think mixing up all the three somewhere. I have to read this concept.
Okay, no issue. Aparna, have you heard about the DNA polymerase, RNA polymerase, and reverse transcriptase?
That I have studied.
Okay, so DNA polymerase and RNA polymerase, these are the two key enzymes used in the molecular biology, right?
Yeah.
And
Yes. Yes.
they have a very significant role in the molecular biology.
Mhm.
Can you tell me the difference of their working mechanism, how they differ from each one? RNA polymerase
DNA
and
poly.
DNA polymerase.
Yeah. So DNA polymerases are basically those enzymes that catalyze the polymerization reaction, or we can say the replication of the DNA, whereas RNA polymerases are involved in the transcription, wherein they are making use of DNA to transcribe into RNA. This is the major difference between the
No, no.
two.
My question was the working mechanism. I know that the RNA polymerase will synthesize RNA molecule and DNA polymerase will synthesize DNA
Okay,
molecule.
working
What is the
mechanism.
working mechanism? On the basis of working mechanism, is there any differences or both are the same?
For this, I'll try to attempt because of whatever knowledge I know I have right now. So DNA polymerase, as we know, it adds DNTPs. So DNTPs are dideoxynucleotides. So it will be having like on second position, like it will be having the hydroxyl group. So based on that, there will be nucleophilic attack like 3 dash to 5 dash polymerization reaction will take place, and it will keep on adding the nucleotides
RNA
that
polymerase will also synthesize in the 5' to 3' directions.
but it
It
will
happens
be
also.
adding like RNA polymerase working mechanism. I think the initiator factors are different. The transcription factors required for RNA polymerases are different, and for DNA polymerases are different. And also the raw material required for both of them are different. I just have to get
Okay,
this.
it's okay. Aparna, tell me one thing. DNA polymerase requires RNA primer or DNA primer for the initiation, right?
RNA prime.
RNA primer. Does also RNA primer use RNA polymerase uses a primer for the initiation?
No,
Why?
because RNA. The reason behind is in DNA, primers are not used on both of the strand. There is leading strand and the lagging strand. The RNA primers are basically used for the lagging strand, wherein the polymerization is not smooth and the Okazaki fragments are produced. So on in
You
that
mean you
region,
mean
primers
there is no use of a primer for the leading strand?
they will be useful. Otherwise,
How
how
it is
will it?
possible that both strands synthesized by DNA
Yeah,
polymerase and the
it
DNA
will be.
polymerase has same kind of activity? And
Yeah, it will be
how it
used
is
for both.
possible that the leading strand DNA polymerase will not use primer and lagging strand will use RNA or DNA or RNA primer?
Definitely, it will be using primers for both the strands. So, why RNA polymers do not need primers is
Okay, if I asked you why DNA polymerase need primer, then
to initiate, like the main idea behind the polymerization reaction is like it keeps on adding the nucleotide to the existing chain. So what happens is they need a free existing 3 dash OH group is there, so that a 5 dash phosphate group of the incoming nucleotide can come and a phosphodiester bond is formed. And in this way they keeps on adding the nucleotides and result in synthesis of entire DNA. So this is the basic idea that I know behind the polymerization reaction. But why, as to why primers are not required for RNA polymerase, this is something that
okay,
I never
leave
got.
it. Parna, can you elaborate the enzymology of a DNA replication in a brief? That means naming and with short function. What are the different
Like
kinds
in
of enzyme used in the DNA replication?
okay, different enzymes. So number one is DNA polymerase. So there are three types of DNA, five types of. Are you talking about eukaryotes or prokaryotes, or it
Just
should
only
be
for
like?
prokaryotes. It will be easy for you. Yeah.
So it's like five types of DNA polymerases are there in prokaryotes as well. One, two, three, four,
Prokaryotic
five.
cells have five different kinds of DNA polymerase.
Like namings were one, two, three, four, five, and what I read is in eukaryotes it is alpha, delta, gamma, and that. In prokaryotes there are three types of DNA polymerases. Yes,
Okay.
three types of DNA. If I'm not now I'm
So
confused.
what is the function of these three type of DNA polymerase?
And so the DNA polymerase 3 is primarily involved in the polymerization reaction, whereas 3 dash to 5 dash exonuclease activity, that is the proofreading activity. Another in polymerases are also involved in the proofreading activity, like in the 3 dash to 5 dash direction or in the DNA repair mechanism. So these have different functions. Other than these enzymes, another enzyme is helicase that unwinds the DNA so that the DNA polymerase can start the polymerization reaction. There are single strand binding proteins that are attached to the open forks so that the helicase that have opened the DNA fork should not again re-anneal. So single strand binding proteins are there to just let the replication fork open. Other enzymes are topoisomerases, like they prevent the supercoiling of the DNA. Other enzymes, DNA ligases that seals the gaps once the primers are removed. Ligases, polymerases, helicases. I think these are the enzymes that are used
What
in the
is
process.
topoisomerase?
Topoisomerase is enzymes that relaxes the DNA, like reduces the supercoiling of the DNA. So, like replication fork is open. It might happen that if from one end single-stranded binding proteins are there and just open the fork. From other end, there might be the case that supercoiling can happen, and it can make the process of polymerization bit cumbersome because a open fork is required so that DNA polymerase can add nucleotides and polymerize the reaction. So it unwinds the DNA and keeps the DNA in position.
Okay, Aparna, suppose cells does not contain topoisomerase, any kind of a topoisomerase. What will happen with that cell?
So definitely there will the problem of the problem in DNA replication will occur because ultimately what I know is it will be causing a supercoiling of the DNA. And supercoiling of DNA means that a polymerase won't be getting enough like a target to add DNTPs. So that only I can recall. It will be a difficult for a cell to undergo the process of DNA replication smoothly.
Okay, Aparna, suppose we have a four different kinds of DNA molecule. That is single stranded DNA, linear DNA molecule, double stranded linear molecule, and a single stranded circular molecule, and double stranded circular DNA molecule. Among these four different type of a DNA molecule, which kind of a DNA molecule will act as a primer for DNA polymerase, and why?
For DNA polymerase. So it will be single stranded RNA molecule.
No, no, all kind of molecules are yeah, single stranded, double stranded, circular, and linear.
All are DNA, but it will be single stranded. It is single, double, linear, and circular.
Yeah, single stranded linear, double stranded linear, single stranded circular, and double stranded circular. All the kinds of molecules are DNA, not RNA.
And you are asking which of the molecule will act as a primer for the process of polymerization.
DNA polymerase. Yeah.
So, DNA polymer. So I think it will be single stranded DNA only because for the activity of DNA polymerase, one end should be free, like existing strand should have a existing nucleotide should have a free 3 dash OH so that the incoming 5 dash can act and form the phosphodiester bond. So 3 dash will provide that thing. And double stranded is the concept of polymerization is like primer will first hybridize with the in existing parental copy of a DNA, and then it will undergo the process of polymerization. So hybridization can only be possible when there are two single-stranded nucleic acids. So that's why a single strand, not double strand, and circular because it won't provide any free end to polymerize the reaction.
Okay. Okay, Aparna, restriction endonuclease is one of the key enzymes used in RD technology, right? So,
Yes.
what is the source of that restriction enzyme?
So source of that is few restriction enzymes are isolated from initially from E. coli and bacteriophages initially, and right now those enzymes can be isolated from various strains. There are
Is it
many
possible to
species.
isolate some restriction enzyme from the eukaryotic source?
Eukaryotic sources, no, I don't think so because I
Okay.
haven't heard any strains.
What is the basic feature of a restriction enzyme?
So restriction enzyme possess two active, basically do two works like either modification or just doing the
No,
modification
no, I
and
am asking about the features, properties of the restriction enzyme. What are the characteristics, properties of restriction enzyme?
characteristic property as in like they are the molecular seizures. They create nicks in the DNA, they recognize the specific sequence in our DNA and then recognize and cleave accordingly based on the restriction enzyme we are using. So these are basically used in the cloning when we want to ligate to vectors and the gene of interest, then molecular seizures are used so that the specific nick can be created and the our gene of interest can be ligated.
What is palindromic sequence?
Palindromic sequences are those sequences that are read same from when we either read from the right direction or from the left direction.
Okay, Aparna, suppose you have a DNA from the plant cells, right? Source of DNA is the plant cells, and you just want to cut that DNA and integrate into some suitable vector, that is positive vector, for for the multiplication of that desired DNA sequences. Will the source of restriction enzyme from the prokaryotic act on the DNA of from the plant cells?
I think so. It can, if the principle behind the restriction enzyme is recognition of the sequences and to cleave it, and if that restriction enzyme can recognize that very same sequence in the plants as well, then I think so they can work because
That means there is no issue. Whatever the source of a DNA, restriction enzyme will act on that.
I think so. It can work, but I am not sure. Like
Okay,
it
what is recognition frequency?
recombination frequency,
Recognition.
recognition frequency.
Yeah.
Okay, so I have heard this term like for the very first time.
Okay, what is isosomers?
Isosomers are a type of a restriction enzymes that basically are isolated from the two different sources but recognize the same sequence and cleave also at the same site. For example, I remember I just read there is some species SPH1 and BBH1 that are the two isolated from two different bacterial strains but recognize the same sequence and cleave also at the same site. Something CTAGC, so between A and G, both.
Okay, Aparna, we have a different kinds of resistance enzyme we know. That is type 1, type 2, type 3, type 4, etc. So which type of resistance enzyme we will use in the recombinant DNA technology?
We make use of mostly the most common one is type 2 restriction enzyme that
Type
we make
3,
use of. Two.
type
Two.
2. Why?
Yeah. So firstly, because it cleaves the recognition sequence, like within the recognition sequences. Other enzymes cleave either away 1,000 base pairs or 10 to 20 base pairs, but type 2 recognition sequence cleave within the sequence. That's
Okay.
why I think that and also it do not utilizes ATP, so it is kind of saving on energy. Other enzymes are utilizing ATP and then cleaving, making cleavages.
Aparna,
Like type one.
okay, got it. Aparna, have you heard about the first line, second line, third line, and last line of a defense?
Yeah, I have. Yeah,
So
first line of.
what is the mean of a last line of a defense?
I think so. This is my just a kind of guess. I think last line refers to the adaptive immunity. If I may not be wrong. First line of defense is something.
Suppose make it easy. Suppose there is an extra cellular antigens encounter in our body.
Actually,
In that case,
yeah.
what will be the last line of a defense?
Extracellular antigen, I think so. Under that, adaptive immunity will come into play
Adaptive immunity.
because because first line of defense is skin and the barriers. Second line of defense is that all the secretions secreted, like I think so, the secretions from the gut or the acid in the stomach. That is the second line of defense. Third line of defense is when cellular barriers come into play, for example, the phagocytes, macrophages, and all. And the fourth line of defense is when these antigen presenting cells will be activating all the T cells, all the MSC complexes will come into play, or depending on the antigen, different complement system that is even a part of innate immunity. But I guess fourth line of defense is somewhat related to the adaptive immunity, wherein I think so.
Okay. Suppose a person's got a encounter with the antigens. So antigens, right?
Mhm.
Which kind of antibody firstly will form in that organisms or individuals?
So, B cell will firstly be producing this IgM only, that will
IgM.
be on their surface, IgM on their
Okay.
surface, and depending on whatever the antigen properties, they will secrete other, like express the other plasma cells to secrete different types of antibodies. First one will be IgM and IgD, like these two are present on the surface of the B cell, but if suppose the cell is like parasitic worm, then they will guide uh, eosinophils and basophils will be releasing and they will activate the IgE, like antibody production. They will guide the B cell to produce IgE. So that way, but the first one will be IgM and IgT that will express on the surface of B cell.
Aparna, how many different types of antibodies are there in our body?
Five.
Five. Can you name?
IgM, IgG, IgT, IgE, IgA.
What is the function of IgD?
IgD is basically that triggers eosinophils and basophils to degranulate and secrete histamines and take mainly participate in the allergic reactions.
IgD, not E.
Uh, Basically, one is responsible for degranulating the eosinophils, but IgD also plays important role in degranulation of basophils
Activation
and
of basophil,
activation of
and basophil
basophils.
involved in the allergic reactions.
Not exactly allergic reactions for that. Ig is mainly responsible for allergic reactions, but IgD somehow all like activate on the basophils and produces degranulations. Like it also play important role in activation of basophils. This is something I read somewhere.
What
I
is
may be wrong.
complement system?
So complement system is basically a part of an innate immunity, wherein it basically uh, is a kind of is basically like works in a cascadic manner, wherein one protein signals the other protein activation, and when a complete set of protein is synthesized, they can be used for certain processes. Like there are three types of complement systems: alternate pathway, classical pathway, or mannose binding lectin pathway. So in all these three different pathways, different proteins are involved, and they work differently. For example, the classical pathways and all the three pathways are involved in the formation of membrane attack complex, which creates the pores in the bacterial membrane so that lysis can occur. Some proteins like C3 proteins are also even involved in the process of, if I remember that, C5, C4, C3 are involved in the process of opsonization also, or some are involved in presenting the antigen antibody complex to the natural killer cells. So complement system is basically wherein it mediates the killing of the antigens by presenting antigen antibody complexes.
Is there any role of antibody in activation of complement system?
Yeah, antigen antibody complex definitely activates the complement system.
So among these different five molecules of antibody, which molecules have a highest affinity to activate the complement system?
IgG and IgM.
IgG and IgM. Why?
The answer of why I don't know. Very honest. Yeah, I don't know why.
Okay, no issue.
Maybe I can remember. I maybe the guess that I is IgM being pentameric in nature can cross-link the antigens and present to the uh, like higher affinity for the antigens and may present it to the that, and IgG being present in the highest amount in the body, 50 to 60%. So it will be keep on circulating in the blood, so maybe recognize the antigen like
Good one.
at a very better pace than others.
Okay, Aparna, suppose you are dealing with antibody M and
Hmm.
go for the SDS gel electrophoresis. Then after the completing your electrophoresis, you go for the gel down. So how many different bands will be there for the antibody M?
I think two bands will be there.
Two
One for
bands.
heavy polypeptide and another one for uh, light chain polypeptide.
And what will be for the antibody G?
For G, it will be again two.
Again two. And
Yeah,
antibody A?
three
Three,
because of J chain. Because of J chain.
because of J chains.
J
So
chain because J chain.
does antibody not no have any chain? J chain?
Which antibody?
Antibody M.
That is what I was. I am just kind of recalling whether all the five antibodies are bound together by J chain. But I think only A antibody has J chain. I have to look at it.
Okay, Aparna, well done. Good for your interview and mock interview also. So right now the session is open for you. If you have any query, any questions, whatever you want to ask to me, then you can ask to me. And now the session is open for you, and you can talk in Hindi also. Feel comfortable and be relaxed. And before going ahead, I will answer the questions which whatever I asked to you. Like as suppose I asked to electrophoresis for the antibody M. So there will be
Hmm.
three band because
Okay, three bands.
one band for all the kind of a light chain, one band
Hmm.
for all kind of a heavy chain, and one band for the J chain because J chain is also peptide. And
Okay, so it
for
is present in
yeah,
both.
and for antibody G there will be only two band.
Hmm.
And if I ask to antibody A, an antibody A has two types. That is one is the secretory antibody and one is the serum antibody. Serum
Okay.
antibody has divalent and J chain. So there will be three band. But if we consider, sorry, secondary antibody contain J chain as well as an extra chain, peptide chain, right? So
Secretary
there will be
peptide.
four band for the secondary antibody. One for the
Okay.
light chain, one for the heavy chain, one
Okay.
for the J chain, and one for the
Okay.
extra peptide chain. And for the serum antibody G, antibody A, that there will be three band: J chain,
Okay. So
light chain, and heavy chain.
okay.
Okay. ओके अपर्णा, आपका इंटरव्यू काफ़ी अच्छा रहा, आपकी प्रिपरेशंस ठीक है, बट थोड़ा सा और अच्छे से आप ब्रशअप कर
हम्म।
लीजिएगा। जैसे अभी आपने कई सारे सवाल जो मैंने आपसे पूछे, लाइक एज मॉलिक्यूलर बायोलॉजी से, जैसे मैंने आपसे पूछा कि मॉलिक्यूलर बायोलॉजी में कि डिफरेंट टाइप्स के जो पॉलीमरेजेस हैं, लाइक एज डीएनए पॉलीमरेज, आरएनए पॉलीमरेज और रिवर्स ट्रांसक्रिप्टेज एक्सेट्रा, इन सब की जो फंक्शनलिटी है वो कैसे डिफर करती है, तो आपको आइडिया है, बट थोड़ा सा मुझे लगता है कि आपको हल्का सा ब्रशअप करने की जरूरत है।
हाँ, कंफ्यूज।
बेटर होगा कि आप एक बार उसको देख लीजिए कि क्या चीज़ें हैं और क्यों ऐसा हो रहा है, राइट? और साथ साथ में ज़्यादा स्ट्रेस लेने की आपको ज़रूरत नहीं है। अभी आप जितना, आप कब है आपका इंटरव्यू?
15th।
15 को,
I
ओके।
have
तो आपके पास अभी
2 days।
अभी आप कहाँ, मेरठ में हैं?
नहीं नहीं, एक्चुअली राइट नाउ आई एम इन जमशेदपुर, झारखंड। सो
झारखंड?
माय हस्बैंड, या बिकॉज ऑफ माय हस्बैंड
अच्छा,
लाइक।
ओके। सो मतलब आपको झारखंड से मूव करके जाना है न्यू दिल्ली की तरफ, राइट?
हाँ, दिल्ली, आई विल बी गोइंग दिल्ली या ऑन
तो
13th April।
ओके, विद इन 2 और 3 डेज आपको क्या करना है, इम्यूनोलॉजी एंड मॉलिक्यूलर बायोलॉजी एंड रिकॉम्बिनेंट डीएनए टेक्नोलॉजी, इन तीनों टॉपिक्स के, जैसे मैंने आपसे बोला कि कौन कौन से डिफरेंट काइंड्स के एंजाइम्स हैं जो कि डीएनए एप्लीकेशंस में इन्वॉल्व हो रहे हैं, मेक अ लिस्ट जस्ट ओनली विद द फंक्शंस, लाइक एज अ फॉस्फेटेज, लाइक एज काइनेज, लाइक एज इंडोन्यूक्लियेजेस एक्सेट्रा, इन सब का एक लिस्ट बना लीजिए, ताकि आपको ब्रीफ करने में आसानी रहेगी। प्राइमेज का फंक्शन क्या है, राइट? टोपोइसोमरेज का फंक्शंस क्या है, और इंडोन्यूक्लियस का फंक्शंस क्या है, इस तरीके से आपको एक आइडिया रहेगा। दूसरा, रिकॉम्बिनेंट डीएनए टेक्नोलॉजी के केसेस में आप एक लिस्ट बनाइए वेक्टर्स का, लाइक एज प्लाज्मिड, फास्मिड, काउस्मिड, लैम्डा फाज, बैक, यैक, मैक, जितने भी वेक्टर्स हैं उनका एक लिस्ट बना लीजिए।
That I have
या
studied, yeah।
उनका क्या कैरिंग कैपेसिटी है, कितना वो होल्ड कर सकते
Mhm.
हैं, किस कंडीशन में आप उनको यूज करते हैं, ये एक आपको ब्रीफ आइडिया होना चाहिए। स्पेशली प्लांट फैक्टर, एनिमल फैक्टर एंड प्रोकैरियोटिक फैक्टर, इनको लेकर के। और रेस्ट्रिक्शन एंजाइम की प्रॉपर्टी मैंने आपसे पूछा, तो आप थोड़ा सा उस पर आपका कॉन्फिडेंस नहीं दिखा। जैसे रेस्ट्रिक्शन एंजाइम की जो प्रॉपर्टी होती है वो ये होता है कि इट विल वर्क इंडिपेंडेंटली, वॉट एवर द सोर्स ऑफ डीएनए डजेंट मैटर, इट मैटर जस्ट ओनली फॉर द सीक्वेंस ऑफ दैट पर्टिकुलर रिकॉग्निशन साइज। जो भी रिकॉग्निशन सीक्वेंसेस होगी उससे उसको मतलब है, ये फर्क नहीं पड़ता है कि वो प्लांट का सोर्स है, बैक्टीरिया का सोर्स है, या
Yeah।
फिर एनिमल का सोर्स है। दैट्स व्हाई वी आर यूजिंग फ्रीक्वेंटली फॉर द जीन मैनिपुलेशन इन प्लांट, एनिमल, फंगस
Mhm.
एक्सेट्रा। सो उसकी सबसे अच्छी प्रॉपर्टी यही है कि वो इस बात पे डिपेंड नहीं करता है कि वो बैक्टीरिया से आया है, सिर्फ बैक्टीरिया के डीएनए में ही काम करेगा, ऐसा नहीं है, राइट?
Mhm.
रेस्ट्रिक्शन एंजाइम को रेस्ट्रिक्शन एंजाइम के नाम से क्यों जानते हैं, इसको भी आप देख लीजिएगा, उसके पीछे की जो हिस्ट्री है होपफुली आपने पढ़ा होगा,
हाँ।
है ना?
हाँ, बैक्टीरिया में वो अर्बल।
तो आपको आरडीटी टेक्नोलॉजी में वेक्टर्स को पढ़ लेना है, ठीक है, उनके कैरिंग कैपेसिटी को लेकर के। और मॉलिक्यूलर बायोलॉजी में तीन जो प्रोसेस है सेंट्रल डोग्मा के, दैट इज पूरा जीन एक्सप्रेशन का, ट्रांसक्रिप्शन, ट्रांसलेशन एंड एप्लीकेशन, इनका जो ब्रीफ है उनको अच्छे से स्टडी कर लीजिएगा। इम्यूनोलॉजी में जो आपने पढ़ा है वो ठीक है, बस एक बार ब्रशअप कर लीजिएगा पूरे एंटीबॉडी को लेकर के, एंटीजन को लेकर के, हाइपर सेंसिटिविटी को लेकर के। ये सारे कुछ कुछ टॉपिक होंगे जिनको कि आप अगर एक बार आप फिर से देख लेंगे तो ज़्यादा बेहतर होगा, ठीक है? बाकी और ज़्यादा कोई ज़रूरत नहीं है किसी चीज़ को करने के लिए, आप के पास अभी जितनी भी इंफॉर्मेशन है वो ठीक है, उसके बेसिस पे आप कॉन्फिडेंटली वहाँ पे इंटरव्यू आप दीजिए, अच्छा होगा, कोई दिक्कत नहीं, ठीक है?
Just
कैसा
one query,
कैसा
sir.
लग रहा अभी आपको इंटरव्यू दे करके मॉक?
So I'm feeling bit confident that I am able to frame the lines and the questions, because that even my husband was asking me questions, so I was fumbling a lot. So he was like, "You are preparing, but you are not confident." So this is something that I realized ki I am able to frame lines now. The just one query, sir, I am having is I'm not confident about my master's project. So like you asked me my intro, right? And it was like I didn't keep my intro open ended so that you can pick questions from my intro. It's just because I'm not confident about my master's thesis. So is it right if I do not say in my intro?
Suppose if they offer the questions like what is your favorite topic, then definitely you have to opt some topic. Or you can say like that ki, suppose, sir, I prefer to go for any subject or any topic. I am not especially for, I am not interested in any particular kind of a subject. I can happy and I can be confident on all the kind of subject. So like that, but it will be very broad sense. Suppose if you offer such kind of answer, then definitely you have to study genetics, cell biology, molecular biology,
Hmm.
microbiology,
It's
and a huge number of topics are there. So that is why it
yes.
would be better if you state that the subject like immunology, molecular biology, and recombinant technology. So you can say like that, sir, I am little bit confident on these three topics. I will try to answer if
Hmm.
you ask the questions from these two topics. So it will be better. So definitely you have a concept about the immunology and molecular biology, but there is a need of a brush up, right? But there is a
Yes.
need of a brush up. So you have a nearly 2 or 3 days time. So in between 2 or 3 days, you just prepare some short statements, like as a list of a vector, like as a cells involved in the immune responses, different kinds of cells involved in immune responses. What is the what is adaptive immunity? What is the innate immunity? Right? Adaptive immunity has further two branches, like a cell mediated and that is humoral immunity. What the different kinds of cells are
Hmm.
involved in the different kinds of adaptive immunity, right? What is the different kinds of hypersensitivity? What different kinds of antibodies are involved in the hypersensitivity? What is the different kinds of vaccines are there possibly? What is their significance? What is the advantage and disadvantage? Comparative analysis about the vaccine, different kinds of vaccines. So questions will be like that. So feel confident, just prepare all those the topics which are very highlighted, like as vaccines, hypersensitivity, immune responses, antibody structures, antibody electrophoresis pattern. And in molecular biology, just go for the DNA replication, transcription, translation, gene
Okay,
expression,
sure.
and certain basic concept about the, it's like as nucleotide structures, and what the different kinds of chemical bonds are there in the nucleotide structures, and how DNA polymerizes in the 5' to 3' directions, etc. So it will be good for you.
Genetic disorders also, na, maybe because I
Genetic
think some of
disorder
the
might be possible, they can ask, but it is not, because genetical disorder is the concern of a genetics, and you are interested in
hmm,
molecular biology. Meanwhile, there is no any boundary for the interviewer. They can ask any question from any side. Okay, so
anyway, yeah,
it
definitely.
can't be predicted. But especially you just focus on these topics, that is molecular, immunology, and RDT, and this will be good. And just go for the brush up and revise your whatever the content you have studied during your mains preparations. No need to be
Hmm.
worried and don't be panic. It's everything is fine. It happens with everyone. No one is
Yeah.
perfect and no one is a master of all the subjects. Okay, so interviewer will also face some kind of a situations if I take the interview of that interviewer. Okay.
Yeah, it might be the case.
Okay, so feel like that. So there is no issue. It happens
So,
with everyone. Suppose if they ask 10 questions, it might be possible that you will able to answer 6 to 7 questions. So definitely the ratio of answering and non-answering question is 70 to 30% most probably. So it is good.
are you, since you are connected with lots of students and students might be contacting you, so did you find any repetition of questions in any of the questions asked
Yeah,
from different
the
students?
repetition of question was that is the your area of interest, your favorite topic, and what the subject you don't like, and right, what are you currently doing, and what is the experience of your current work, what will you do after suppose you are not getting selected here. Such kind of questions are repeated for everyone. So be prepared for that.
And
These questions are
technical
actually
questions.
these questions are very patent. Like the what
That
you're
is
currently doing. Those some questions will from ask from current work. So it's obvious and it's a journal.
so, and one question I have, that you have asked from someone in during the mock interview only. I'm just inquisitive about the answer. You were like DNA polymerase is the enzyme, but how it can bind to four different DNTPs. So that was something I was searching till that day, but I didn't find a crisp and the precise answer. So can you help me with that?
Yeah, definitely. See, DNA polymerase is an enzyme which just only add the different kinds of nucleotide, not polymerize. It do not
Hmm.
have ability to initiate the polymerization process. If you look the molecular mechanism of a DNA polymerase, just there is a active site for the DNA polymerase, and it has a metal by metal, that is magnesium ion, right? And why it
Yes.
requires 3 prime or why it requires primer? Because primer have a 3 prime hydroxyl group, and that
Yeah.
3 prime hydroxyl group will attack on the upcoming nucleotide and will remove the double phosphate from the
Okay.
ATP molecules, and the
Yeah.
DNA polymerase will act only to the addition of that nucleotide. So for the addition, there is no need of a substrate molecule will go inside the active site and bind for that particular kind of a substrate molecule. That is why DNA polymerase will able to utilize
Got
four different kinds of substrate. Actually, these are the
so
substrate, but DNA polymerase is not binding nucleotide molecule in their active group. Is it just
so that
only linking the bond?
okay. So that is the reason why it is unable to polymerize in 3 5 prime to 5 prime direction also, because
Exactly.
single phosphate is
Yeah.
there. That's why.
So all the questions,
Okay.
all the answers will be here. You can just look forward. Suppose
Correlated.
why DNA polymerase always synthesize in 5 prime to 3 prime directions. It can be answered from this concept. Why there is a need of a primer? You can just answer of these questions. Why DNA polymerase can use four different kind of polymerase, different kind of nucleotides? So that is the reason behind is that is there is no binding of substrate molecules inside the active site of a DNA polymerase. DNA polymerase just adding the nucleotide by forming a phosphodiester bond.
Oxidized form. And why RNA polymerase do not require primer? Like this is
Very good.
something.
Think about
This is
it.
some.
I have given a clue for you. Think about it and just try to find your find answer on the behalf of you.
Huh,
If you
I'll
are
find
unable
it.
to find, then definitely I will answer you. You have a
Huh.
time 2 or 3 days.
4 days.
You
Yeah,
can
yeah, I
just
have.
message me.
Yeah, sure.
It will
And
take
also, I'm not confident about it. So if you don't upload, then it will be good for me. I'm little less confident. So it's like I thought of like first you
definitely you
can
will
send me.
definitely if you find the answers of different kind of questions itself, it will boost your confidence, right?
Hmm.
So if there is no any sources and you are not able to find, then definitely I am here for you. You can just message me, I will explain.
Mhm. Definitely. I'll
Okay.
just try searching on Google and see if that explains me
Just you
more.
have to think about it and analyze all the concept whatever you have about the DNA polymerases, RNA polymerases. If you more analyze and if you more think, then definitely you will get answer. Google cannot explain each and everything.
That is true.
Okay. So many things.
That is very
Yeah.
true.
Okay, Aparna.
Thank you
Nice,
so much.
nice to
Thank
interact
you.
with you, and hopefully you are also enjoying this interactive session and
Hmm.
feeling good right now.
Yeah, yeah. I love these kind of questions when it just strikes our
Don't
mind.
worry, I will share the recorded video lecture, and
Hmm.
just watch this video again and again, and write down the whatever the questions I have asked to you, and try to find and try to write the answer of that questions.
I
Okay.
am doing this from past all the interviews you are doing. I am writing questions. I am finding answers. I
Oh
am
my
just.
God. That means
I
now you
just
will have a, bunch of questions, right? Collections of questions.
yeah, yeah. Like I can tell your questions, PGPRs and all PSBs, your love for plant biotech and
कैसा
everything.
लगा ये क्वेश्चंस जितने आपने वीडियोस को देखे हैं उन सारे वीडियोस में जो क्वेश्चंस पूछे गए?
It's like quite a brainstorming. Like for a student of biotech, this is something we should know. Like this is a very minimal expectation that a student, as a student, we should like complete.
कभी जो जो सवाल जो क्वेश्चंस आप लोगों से वीडियोस में पूछे जाते हैं, लाइक आज आपसे पूछे गए और बाकी बच्चों से पूछे गए, क्या ऐसे टाइप के क्वेश्चंस को पहले कभी ऐसे सोचने का मतलब पॉइंट ख्याल आया था या फिर कभी ऐसे सुनने को मिला था?
वैसे सर आईआईटी बॉम्बे की ये खास बात है कि प्रोफेसर्स ने वाकई ऐसे-ऐसे क्वेश्चन से हमको हिलाया ना कि वो सोचने की क्षमता कि ये क्यों नहीं सोच रहे तुम लोग? ऐसा सोचना चाहिए। वो चीज़ तो वहाँ से आई कि तभी अब से ऐसा होने लगा हर क्वेश्चन से पहले व्हाय तो आता ही है, व्हाय। दैट इज़ समथिंग वी हैव लर्न्ड फ्रॉम दैट मास्टर्स कि व्हाय तो होना चाहिए। हो सकता है हर एक का व्हाय ना हो बिकॉज़ बायोलॉजी इज़ समव्हाट not everything can be experimented right now. Some are fact based also, but you should have that why every time. So this is something. Now when you were saying this, so I was discussing, so she was like, यार ये तो कभी सोचा ही नहीं, polymerase enzyme है और 4 न्यूक्लियोटाइड्स होते हैं, फिर वो भी आईआईटी बॉम्बे से वो
Have
भी
you
सेंड।
discussed such kind of questions with your friends?
Yeah, from like my few of my friends from IIT Bombay are just with me only right now. So we were just discussing, we were having a good discussion. So we were like, प्रोफेसर ने बताया था, देख हम भूल गए। मैंने कहा ये अभी याद करना है।
चलिए,
So
ओके।
yeah,
होपफुली कि
that
जितने
was
भी रिकॉर्डेड वीडियोस यूट्यूब पे अवेलेबल हैं उनसे काफ़ी हेल्प मिल रही होगी, जैसे आप बता रही वैसे बाकी लोगों
definitely।
को मिल रहा हो। और यही
नहीं नहीं
पर्पस है उस
definitely।
वीडियो का कि बाकी लोग देख करके भी समझें कि हाँ, इस तरीके के सवाल आपसे बन सकते हैं और ऐसे सवाल का आंसर आपसे उम्मीद की जाती है, बीइंग अ मास्टर्स ऑफ बायोटेक्नोलॉजी, राइट? सो
Hmm. Definitely these are required।
ओके
Thank you
अपर्णा,
so much।
so
Thank you।
wish you all the best for your for your interview from my sides. If you feel any kind of trouble or any kind of a questions or doubt, then free feel to ask me. You have my number, and also you are a member of that group, so you can message me.
Yeah, yeah,
Definitely, I will be there
yeah।
for you.
ठीक है, थैंक यू सो मच सर।
Okay.
थैंक यू।